We've been talking about prokaryotes. We've already gone through archaea, and so now we're going to switch over to bacteria. So bacteria are a very large, diverse group of prokaryotic organisms. They're also a very large, diverse group of organisms on Earth. So we mentioned that between bacteria and archaea, they take up about 50% of all the biomass on Earth. So there are a lot of organisms. There's a wide variety of organisms, but we don't have the time in this class to even scratch the surface on the complexity and the diversity of these organisms. However, we are going to talk about two really important ones, cyanobacteria and the proteobacteria. And that is because these two participated in the endosymbiont theory, or endosymbiosis, that led to the formation of our eukaryotic organisms. Now, this is important because we keep talking about how we are going to work our way through organismal diversity. But these guys are also important in their own way. They also provide some very important services. They're a very important groups of bacteria. So molecular studies suggest there are about 100 bacterial phyla or 100 bacterial groups, and then all of those groups are broken down into much smaller categories. Kind of like we have our eukaryotes, and we're going to get to our vertebrates and then all the way down to mammals. There's a whole bunch of other categories in there, but if you think about the diversity of just animals, and then you consider that all of those bacterial groups that I showed you, plus many, many more, contain just as much diversity. So cyanobacteria and proteobacteria are especially diverse and important. We're going to talk about those guys. If you are interested in any of the other bacterial phyla, the very big major categories that I showed you in the previous slide, you can check out table 23.1 in your textbook. Just kind of gives a brief description of what they are and why they're important. If you are more interested than that, then Biology 270 in our biology department is our Biology 250 is our microbiology class, and you'll go through a bit more of bacterial importance and biodiversity. So most bacteria are going to favor what we would call moderate or normal conditions. We talked about how archaea are very often extremophiles. There are some bacteria that are extremophiles as well, and those bacteria are going to have very special adaptations for those extreme environments. But most of our bacteria that we're going to talk about live in sort of normal environments where we would encounter them or we would be familiar with them. We mentioned earlier that bacteria are found in nearly every habitat found on Earth. So anything from the doorknob in your bathroom to your hands, your skin, in your intestines, on your dog's paws, whatever you could think of, there's probably bacteria that live there. Now, most of them are actually completely harmless to humans, completely harmless to other organisms. There are absolutely some that are pathogens that cause disease, that cause us problems. But most of them, and in fact many of them, form actual Most of them are harmless. Many of them form symbiotic relationships with eukaryotes. So we have a whole slew of gut bacteria that are helpful in helping us break down our food that we can't break down very well, help produce vitamins for us, help regulate our system. There's all kinds of evidence that say that your gut health is linked to many other aspects of your health. So there are a lot of bacteria that have symbiotic relations with us and many other eukaryotic organisms. So cyanobacteria are important because they are a photosynthetic bacteria. Now, we talked about them as the precursors to our chloroplasts, and how they are able to perform photosynthesis. So they are the bacteria that was engulfed by an ancestral archaea and then evolved that symbiotic relationship with the archaeal cell and was able to use its photosynthetic abilities to become a photosynthetic organelle. So they're named cyanobacteria because they appear to be green, blue-green, cyan, which is a pretty blue color, in their cells. So we're going to look at some of these in lab. They're going to actually look the color of plants, but they do kind of range from like a blue-green to a green color. So these photosynthetic bacteria are usually found in water. They can be found in pretty much all of our aquatic environment, so freshwater, oceans, wetlands. They're even found on soil surfaces. And they are important because they produce oxygen. So just like plants produce oxygen, and they do this by taking in sunlight energy, converting that sunlight energy into glucose, which is the main purpose of photosynthesis, that glucose production and energy storage. But as a byproduct of photosynthesis and that glucose production, they generate oxygen. So oxygen is released during all photosynthetic processes, whether they're plants or cyanobacteria, algae, whatever they might be. So prokaryotes, along with plants, are really important in generating oxygen that the rest of us require for metabolism and cellular respiration. So you breathe in oxygen during the process of cellular respiration. You breathe out carbon dioxide. And then those plants and bacteria take in that carbon dioxide, produce glucose. So they're an important source of oxygen on Earth. Plants take up about 50%, maybe more, of our photosynthetic processes. We also have other organisms, some other eukaryotes like algae that perform photosynthesis, and then we've got these cyanobacteria. So we've got a lot of organisms doing it, but cyanobacteria do a lot of it. They're able to do this through specialized membranes called thylakoids. Now you might remember when you took Biology 101 that you had this structure that was the chloroplast, and then there were these stacked structures within that chloroplast that were called thylakoids. So the chloroplast is an organelle inside eukaryotic cells. Cyanobacteria are prokaryotic, which means they don't have those organelles. So they don't have a chloroplast, but they do have the structure that you would find in the chloroplast, which are these thylakoids. Now in these thylakoids we have chlorophyll, and that chlorophyll collects the sunlight energy. So these thylakoids in a bacterial cell are going to be folded pieces of membrane inside that bacterial cell. That's going to have exactly the same function. Now this is why we get those bacteria, the cyanobacteria, dissolving into the chloroplast, because they already have the structures inside them. Function in eukaryotic cells. So we get this endosymbiotic event where we get chloroplasts inside our eukaryotic cells, and that's going to lead to eukaryotic algae and plants. And we're going to see, once we get into our protist section, that algae are the ancestors of plants. So we've got cyanobacteria that sort of lead the way, the evolution of plant cells. So cyanobacteria are important for oxygen production. And that's a good thing. But they are also responsible for some less than pleasant processes as well. So one of the problems that we get are sort of cyanobacteria blooms. We can also get other types of blooms. This is when an organism basically has a huge population boom, and this causes problems for the environment in which it's in. So usually we see this in water. We can see this for algae, we can see this for cyanobacteria, and we're going to talk about this later when we get to our ecology section. But if there is phosphorus in the water, and there is too much phosphorus in the water, but cyanobacteria can go through this process that we call blooming. And this is where we get huge explosions of cyanobacteria or other organisms, so much so that the cyanobacteria become visible. So you might, if you Google this, you can see like algal blooms, cyanobacteria blooms. They're visible from space. Like they are absolutely enormous. And the problem is that when this happens, the waters become toxic. So basically, the organism that blooms uses up all of the resources very quickly, uses up all of the resources, and then they die. And this produces a toxic environment for everybody else. So all the other plants, fish, algae, etc. in the water are going to die. Now, this happens because of human impact. So when we release phosphorus-rich fertilizers, when manure or sewage gets into the water, this is going to cause those blooms. So very often we get these blooms as a result of agricultural runoff. So farmers apply these fertilizers to the soil, and then those fertilizers wash off into nearby waterways and cause these blooms. So reducing the release of fertilizers, making sure that we're not leaking manure and sewage into our waterways is really important. But cyanobacteria are not all bad. They do produce some antibiotics. They also produce something called mucilage that helps stable sediment. So we'll see what mucilage is in a minute. It's basically just a slimy layer that allows bacteria to stick together. So this can act as a way to stabilize soil, especially in aquatic environments, because you get sort of these mats. And remember, we saw mats of cyanobacteria. Those were the stromatolites. Stromatolites. That were our first prokaryotic fossils. So stromatolites are still a thing. They are an example of these sediments that form by these mats of cyanobacteria. Cyanobacteria can also fix nitrogen. That means pull inorganic nitrogen usually out of the soil and convert it into an organic molecule. So this is important because a lot of organisms, especially plants, require nitrogen, but they can't use the inorganic form. And so cyanobacteria are some of the types of organisms that can pull in that inorganic nitrogen and convert it into something that is usable for other organisms. Cyanobacteria are also interesting because they exist in a lot of different sort of cellular formations. So they are single-celled organisms. Sorry about that. They exist in single-celled organisms, but they do exist in these different formations of groups of single cells. So you can find cyanobacteria as single-celled. So here's an example, a picture there. You can also find them as colonies held together by that sticky mucilage. So this is that sticky outer layer. And it kind of acts like a glue to hold those cells together. So we've got that example here, our colony of cyanobacteria. That's how we're going to get our mats. You can also see them as sort of filaments, so long chains of cells, as either unbranched filaments or branched filaments. And in lab, we're going to look at an organism called Anabaena. Mentioned this one already. This is a cyanobacteria that forms these long chains of filaments, usually unbranched. Our next important group are the proteobacteria. So these are important because they're related to the ancestors of our mitochondria. So these are ancestral bacteria that can use oxygen to produce or to release energy. So in the process of cellular respiration, glucose is broken down in the presence of oxygen, and that oxygen is used to power the production of ATP. So this isn't the only thing that's important about proteobacteria, but it is for eukaryotic cells because this is how we get our mitochondria. As their own group, proteobacteria have several smaller subgroups. These include a whole bunch of different species that are important for medicine, for agriculture, for industry. There are many that cause disease. E. coli, that's our picture here, is an example of a proteobacteria. So this guy is found normally in our guts. Sometimes E. coli can cause disease. So if you get food poisoning, it's likely because of a pathogenic E. coli that was present on something that you ate. So these guys are important. They don't produce oxygen. They actually use up oxygen. And these proteobacteria can use it to consume methane. They can use it in a variety of different ways to produce energy. So, sorry about that. These guys have the potential to help us to reduce the methane production that is important for climate change. So, some of our organisms, like there are some bacteria, there are many archaea that produce methane as a byproduct when they break down organic materials, and proteobacteria actually consume methane. So there is the potential, not being used at the moment, but the potential to use proteobacteria as a way to remove methane, which is a very, very powerful greenhouse gas involved in global warming, actually help reduce the effects of climate. So a couple of quick examples of proteobacteria. These are all kind of awful ones that cause terrible diseases. We've got Neisseria gonorrhea. This is a sexually transmitted infection that's caused by a proteobacteria. We've also got Vibrio cholera. So this is responsible for cholera epidemics that cause really bad intestinal problems. And then we've got Salmonella and Escherichia coli. These are our common bacteria that give us food poisoning. So all bacteria reproduce in roughly the same way. They can reproduce very quickly. So for example, in optimal conditions, E. coli can reproduce every 20 minutes, which means they double their population every 20 minutes. So from a very small number of cells to a very large number of cells in a really, really small amount of time. You all learned about mitosis in Biology 101. During the process of mitosis, this is our vertical transmission of genes from one organism to another. You copy your DNA, that DNA is partitioned, and the cell splits. Now, bacteria reproduce in a couple of different ways. They can reproduce asexually by budding, which is basically where you have a parent cell and a smaller cell kind of pinches off of the larger cell. And so you're left with the larger parent cell and the smaller little bud cell. These guys are going to be clones of the original cell. And so some of our bacteria reproduce that way. Most of our bacteria reproduce by binary fission. So binary fission is when a single cell divides into two equal cells. So what happens here is you have your parent cell that's got DNA inside. That DNA is replicated and the cell elongates. So we get our chromosome that is separated. This is kind of a several-step process. but that chromosome separates. We should say the chromosome is replicated. It separates and the cell elongates. And then that cell sort of pinches off in the middle, and we're left with two cells that are clones of one another. So this is not nearly as complicated as mitosis. There's not a set of defined steps. So you may remember learning 4 or 5 steps in mitosis, where the chromosomes are doing all kinds of things and moving around. Binary fission is similar in that we get one parent split into 2, DNA separated, but it's far less complicated. And the separation of that DNA is far less complicated because there's only one piece of DNA.